So yan, to cap yung ating research series dito sa YouTube channel, I will explain or I will show you an example of a scientific paper that I created last semester lang. So dito, I will demonstrate yung may introductions, methods, results, and discussions or yung IMRAD na format na sinasabi natin. So this format, ito yung karaniwan na na format ng mga journals. So if you are reviewing your literatures, ganito yung format ng mga that are being followed by those literatures. So dito sa sa format na to, yung RRL ay nakapaloob na sa introduction, yung mga supporting literatures ay nasa discussions na rin, nasa methods, ganyan. So, you can see different literatures being utilized all throughout the paper as a reference. So, hindi na siya yung parang sa chapter by chapter. Nasa chapter 2, dun mo lang ilalagay lahat ng mga literature mo. Parang gano'n. So, let me share my screen. So, yan. I chose this study kasi I think this will be this will be understandable for every audience, sa lahat ng audience. So, yan. So, phytotoxic evaluation and germination aftermath of vinegar, calamansi, chili, and tomato extract using Vigna radiata seed germination assay. So, parang sobrang complex niya, di ba? But, ano lang yan, it is just a simple tawag dito, ganito lang yan. Sige, i-explain ko muna yung main idea ng topic na to, ng research na to, kumbaga. So, dito, titignan natin yung epekto ng vinegar, ng calamansi extract, ng chili extract, saka ng tomato extract doon sa pagtubo ng mga munggo seed. Yung Vigna radiata, it is the scientific name of munggo. So, we will look at that, kung ano yung magiging epekto niya, kung bang tutubo agad yung mga seeds or it will hinder the growth of the seed. So, ayun lang yung main idea dito. So, this is just an exercise namin sa isang subject namin. So, in college, kapag nag-college na kayo, yung mga research papers dati that we are doing for how many months, ay gagawin nyo na lamang in 1 week, gano'n. Or even days before the deadline, minsan gano'n din kami gumawa. Yan. So, yan. So, it is very important that as early as your, as early as now, you are in high school or senior high or junior high, you are already practicing kung paano magsulat ng research paper. So, lalo na sa mga sa scientific na course. For example, chemistry, biology, yung mga yan. Agricultural biotechnology, yung mga yan. So, you will be utilizing a lot of this research paper knowledge pagkarating nyo ng college. So, yan, let's go. So, this is my title. So, yan, it is just an exercise. It is just a part of our exercise for isa kong subject, isa kong course. Yan. So, yan. So, here, ayan yung abstract, pero, sige, mamaya na natin yan balikan. So, let's first go to our introduction. So, introduction dito, first and foremost, sa first paragraph, we can see na, we can see that yung una is dinefine ko muna yung anong meron dito sa research na ito. The first sentence, available water and oxygen, suitable temperature, and absence of inhibitory substances or conditions that are needed for seed germination. So, ibig sabihin, dinefine ko muna yung seed germination. So, that is somehow the problem of this research. So, dun pa lang, nandun na siya. So, if you will notice, wala ditong background of the study, statement of the problem, yung mga ganon, kasi nakapaloob na siya dun sa loob. Sa introduction mismo. Yan. So, seed germination is run by various hormones that are endogenous. So, here, I tried to explain kung anong meron sa seed germination. First paragraph. Ano yung nakaka-apekto sa seed germination process. So, dito, makikita na natin na binanggit ko na dito yung pH, which is one of the factors that can affect the germination. So sabi ko dito, pH also highly influences the germination. So according to you et al. 2012, so nandito na yung literature, yung studies. So inilagay ko dito, the optimal pH value for mung bean seeds is 7 to 7.5 to obtain high seed germination and seedling growth. So dito, I used a related literature na nagsusuporta doon sa kiniklaim ko na ang pH ay nag-i-influence sa seed germination. So, yun. Then here, sa last na paragraph, actually, di ba? Sobrang ikli lang ng introduction ko dito. So, in this experiment, the effect of naturally occurring extracts such as calamansi, chili, tomato, and other chemical such as vinegar on seed germination was assessed through seed germination assay. So, basically, this is my statement of the problem. This is my objective. To assess yung epekto ng mga chemicals na yun, ng mga extracts na yun sa germination ng munggo. So, yun. So, ayun na yung problem and everything. So, nandiyan siya sa introduction. Ngayon, sa materials and methods, so makikita natin dito, ayan, extracts of calamansi and tomato was obtained through squeezing the fruits. So, dito sa materials and methods, pinagko-combine na natin, imbes na parang the materials were as follows. Parang ganon. What I did here, ay na-mention ko na doon mismo sa methods kung ano yung mga materials. So, yun. Extracts of calamansi and tomato was obtained. So, ano yung mga materials. Available vinegar from the kitchen was utilized for the experiment. Ganyan. So, basically, sa materials and methods, you will just discuss kung ano yung ginawa mo. As what I've discussed din sa aking past video about methodology. Ano yung ginawa mo. And, yun. So, you will use a future tense if you are doing a research proposal. Pag sinabi natin future tense, ito yung will be. The root and shoot length will be. Blah blah will be gathered. Yung mga ganon na mga sentence construction. So, dito, gumamit na ako ng past tense which is were. The root and shoot length were measured using a ruler kasi tapos ko nang ginawa. So, ganun yung gagamitin nyo kapag tapos nyo na yung research nyo. But if you are doing a research proposal, will be ang gagamitin. So, yan. Next is sa results and discussion. So, nandito ko sinagot kung ano yung mga problems ko kanina. Diba kanina sabi ko dito sa introduction, ayan, effect on seed germination. So, dito, inuna kong in-state ang germination rate, root length, shoot length, Viger index, and percentage of phytotoxicity. So, in that way, pinresent ko siyang ganon. Kaya, dito sa ating results, una ko rin ipipresent ang germination rate. Then, susunod kong ipipresent yung root and shoot length. Tapos, susunod kong ipipresent ang Viger index. Then, next kong ipipresent ang percentage of phytotoxicity. Ganon yung order ko when I presented it in the objectives or in the statement of the problem. So, dapat ganon din yung order ko when I present it in my result, in my conclusions, in my discussion. So, yan, tingnan natin. So, here, I used a tabular format. So, table, kasi mas madali siyang tignan. So, kapag tinignan mo yung table, makita mo agad, okay, so sa water, 90% yung germination rate niya. Sa ganito, so, pinakamataas pala yung water, yung control natin. Tapos, ang susunod na mataas is yung chili extract. So, mas madaling makikita kapag sa tabular form. Ngayon, titingnan natin kung paano ko siya diniscuss dito. So, sabi ko, table 1 shows that the percent germination among treatment. Shows the percent germination among the treatment. The control setup gains a close to 100% germination, which means mas marami halos nag-germinate lahat ng sa control setup, which is ayun yung expected natin. Followed by chili extract, calamansi extract, tomato extract, and vinegar, respectively. So diyan, tinignan ko na yung order sinabi ko na yung order ng germination rate. So, pinakamataas sa water, tapos chili extra, calamansi, tomato, and vinegar. Tapos, sinuportahan ko siya dito ng isang literature. So, ginamit ko itong literature na to, yung kanina rin, yung sa according to you et al., the optimal pH value for mung bean seeds is 7 to 7.5 to obtain high seed germination. Knowing that water has a neutral pH, which is 7 to 7.5, it explains why it gained the highest germination rate. So yun, ito yung tinatawag nating cross-referencing. That you are, ino-compare mo yung resulta na nakuha mo sa mga available na researches, sa mga available na studies. So dito, kinumpare ko siya sa study ni Yu et al. 2012. Ayan. So, using this, mas nagkaroon ako ng support dun sa naging result. Sabi dito, furthermore, since vinegar has the pH of 2, around 2.5, it inhibits the germination ID. However, the germination of tomato, pH, blah, blah, is somehow comparable, having a 20 to 25% resistance. So, I just explained kung ano yung mga, yung epekto ng pH ng mga vinegar, ng tomato, ng calamansi, doon sa germination nila. Ayan yung tinatawag nating cross-reference. So, same true dito sa mga iba. Ayan, as seen in table 2, ayan, gumamit ulit ako ng table. Ayan. So, the trend, blah, blah, blah. So, in discussing your result, hindi mo na siya sabihin na in replication 1, ganito yung nangyari. In replication 2, ganito yung nangyari. Hindi ganon. Ang gagawin mo na, observe the trend, tapos ayun yung i-discuss mo. Kunwari, ano yung trend ng kunwari, ano yung pinakamataas, ano yung pinakamababa, ayun yung mga mahalagang ilagay mo sa discussions mo dito. Same through here. So these are also cross-references. According to Grant 2020, the acetic acid in vinegar dissolves the cell membrane, thereby leading to desiccation of feces. So somehow this is a cellular na interpretation or cellular fact para mag-support sa aking finding na yung vinegar ay hindi nag-germinate. Kasi nga, sabi ni Grant 2020, na yung acetic acid daw ay nagdi-dissolve ng cell membrane. Thus, hindi na tumutubo yung mga buto. Same through here. Cross-reference. Yan. So, sa summary and conclusions, ayun, as what I've mentioned din sa aking past video, you will just summarize kung ano yung nandun. So, kanina, if you will notice, inuna ko sa objective yung germination rate, tapos pati sa results and discussions, ayun din yung una kong diniscuss. Ngayon, ayun din yung una kong pinresent dito sa summary and conclusion. So here, inuna ko inilagay ang germination rate. So ang summary ito, yung first paragraph. After the test conducted for this study, it was found out that vinegar is the most toxic. So that itself is a summary ng buong study. Ano yung general findings na useful para sa mga readers mo, para sa mga susunod na magbabasa ng paper mo. So, yun. Ayun siya. So, ito na yung mga conclusions sa bawat sub-problem. Kanina, di ba, inuna ko i-present yung germination rate, Viger index, yung mga yan, plus percentage of phytotoxicity. So, in that order, ganun ko rin siya ipinresent dito sa aking conclusion. Germination rate, sumunod yung Viger index, then yung percentage of phytotoxicity. Yan. Kasi ito na yung mga other explanation. The major extracts, ayan. So, ayan na yung mga nakita kong possible reasons. Lastly, itong literature side. So, we are using APA for scientific papers, and yun. Actually, ayun din yung karaniwan talagang ginagamit. That's all. So ngayon, punta naman tayo sa abstract. Sa abstract, bibigyan natin sila dapat ng So ito una, first paragraph, ito yung parang problem natin. Ito yung naging topic natin sa buong research paper na to. Tapos nandito na yung result. Yan, di ba? Results show that vinegar is most toxic, blah blah. Result. Then ito na yung mga supporting details, conclusions, and summaries sa last na paragraph. So dito, parang walang isa-summarize yung buong paper, isa-summarize mo lang siya in 250 words. So yun, I hope that this example helped you in your research paper, kung ano man yung topic nyo. And just comment down kapag meron kayong question. Bye, like and subscribe.